Hello everyone. Today we're going to be doing our Phage Lab report video. So our group is group number 6 and my name is Garza and my partner is Jazlyn Gonzalez. Starting off with the introduction, bacteriophages or phages are viruses that infect and lyse bacteria, creating visible clearings called plaques on a bacterial lawn. These plaques allow us to measure phage concentration through techniques like serial dilution and whole plate titers, which reduce a highly concentrated phage sample into levels that can be accurately counted. In this lab, we diluted a phage solution from 10 to the negative 1 to 10 to the negative 6 and mixed each solution with E. coli to produce plaque-forming units on LB agar plates. This process helped us observe how phage concentration affects plaque formation and allowed us to calculate phage ineffectivity while practicing essential microbiology techniques such as aseptic handling, correct pipetting, and temperature control. Now for our material. We're starting off with culture. We have 10 to the negative 1 dilution of phage provided by the lab technician, as well as an E. coli broth culture. As for the media, we have sterile water, LB agar plates, 4 in total, and molten top agar, which was maintained in a 45 to 50 degrees Celsius water bath. And lastly, for our equipment, we used micropipettes and sterile tips, as well as 9 microcentrifuge tubes for dilutions and E-labeled tubes, as well as 4 large test tubes, a water bath, a vortex mixer, parafilm, and an incubator. Moving on to the procedure. For the first part, we have serial dilution preparation. So for number 1, we received a 10 to the negative 1 phage dilution prepared using the same procedure we would follow. Moving on, we have 5 microcentrifuge tubes, which were labeled 10 to the negative 1, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6. Moving on, each tube received 900 microliters of sterile water. And then for 4, from the 10 to the negative 1 tube, 100 microliters was transferred into the 10 to the negative 2 tube and was mixed. For the 5th step, this dilution process was repeated down the line until reaching 10 to the negative 6 tube. And for the 6th step, after mixing the final tube, 100 microliters of 10 to the negative 6 dilution was discarded. Now for the 2nd step, which is preparing whole plate titer. For the 1st step, 4 LB plates were labeled with the date, type of phage, as well as the dilution that was used, either 10 to the negative 3, negative 4, negative 5, or negative 6. As for the 2nd step, 4 microcentrifuge tubes were labeled E3, E4, E5, and E6. For the 3rd step, from each corresponding dilution tube, 100 microliters of phage dilution was transferred into the E tube. For the 4th step, 100 microliters of E. coli was added to each tube, leading to a total of 200 microliters. For the 5th step, tubes were vortexed and left to stir for about 10 minutes to allow the phage absorption. For the 6th step, 1 plate at a time, the following steps were done quickly. This was by having 200 microliters of phage and E. coli mixture that was transferred into a large test tube containing 5 milliliters of molten top agar, as well as and then moving on, we had the entire mixture, which was immediately poured onto its labeled LB plate. And then we had the plates that were gently swirled to evenly distribute the agar. For the 7th step, the plates sat undisturbed until the top agar solidified. And for the last and 8th step, plates were inverted, wrapped with parafilm, and placed in the incubator.
Now for our results and observations. Across the serial dilution plates, we observed that there was a clear decrease in plaque density as the phage concentration decreased as well. To begin, on the top left, we have our 10-3 dilution, which showed a confluent lawn and no distinguishable plaques. This indicated very high phage activity. On the top right, we have our 10-4 plate, which displayed numerous uniform well-defined plaques. This made it the best dilution for accurate counting. On the bottom left, we have our 10-5 dilution, which showed only a few isolated plaques, meaning that the phage was still present but at a much lower concentration. And finally, on the bottom right, we have our 10-6 plate, which showed no plaques. This indicated that the dilution was below the detectable limit of phage activity. These patterns confirm successful infection and provide a clear dilution series suitable for estimating phage titer. Overall, this lab successfully demonstrated how serial dilutions and whole plate titers are used to measure phage concentration by observing plaque formation at different dilution levels. Our results showed the expected pattern. Lower dilutions produce overcrowded plates, while higher dilutions resulted in fewer, more distinct plaques that were easier to count. This experiment reinforced the importance of accuracy in pipetting, timing, sterile technique, and maintaining proper agar temperature, as small errors can greatly affect plaque distribution and reliability. By completing this procedure, we gained a clearer understanding of phage bacterial interactions and the fundamental methods used to quantify viral particles in microbiology. For our references, we did use the laboratory manuals and the lab instructions provided by Dr. Nagin. Thank you for tuning in, and we hope you enjoyed.